Alright guys, in this video I'm going to be talking about how we can, given some plasma B cells, figure out which of these B cells actually have a high affinity for a target that we have, such as a tumor cell, in this downstream process. And so if we recall, after I bred a mouse in a bubble and euthanized it after injecting it with tumor cells, I was able to extract 200,000 plus or minus plasma B cells. And so right now I have 200,000 B cells, and they're all producing tons of antibodies. And I want to find which of these 200,000 B cells is making the highest quality antibody, because if my goal here is to make a cancer drug and give you antibodies that'll kill your cancer specifically, I want to know which of these is the best product to give you, which will have the highest potency and not be toxic. And so the first step in this process is going to be actually figuring out how we can determine which of these 200,000 B cells is the best, and we want a handful of them ideally. We don't want to be working with 200,000 B cells because it's very hard to deal with. And what we really care about at the end of the day is not necessarily the B cells. We really care about these antibodies, because we can sequence the antibodies and then develop even farther downstream processes to actually create them. So we almost don't even care about the B cells. We care entirely about the antibodies in order to get this cancer drug to market. And so if we'll recall correctly, so we got our 200,000 B cells after we enriched our mouse's spleen, and this came from taking that extract of the tumor cells from the mouse, and now we've got these B cells. B cells, again, I recommend watching my first video on B cells, but they are very unique, and these unique B cells are going to be cranking out antibodies that may or may not, and most likely they may not, have a affinity for the tumor that we care about. And so generally what scientists do in research labs today is they have characterized the surface receptors on tumors very well, and so what they can do is they can make use of bead-based assays in order to bind to this cancer. Now, I'm going to discuss a slightly different variation to this, and I'm going to try to keep this high level without getting too deep into this, and we're going to stick to a constant story here, just that I have a video that makes somewhat sense to me. So what I'm going to say is there's a company called Berkeley Lights, and they have something called the Beacon and a new product called the Lightning. And specifically on the Beacon, we can use this for antibody discovery, in which we're going to be having individual B cells inside of pens. And so this plasma B cell that I have, the purple thing here, plasma B cell, will be loaded into a pen. So this will be the plasma B cell, and it's going to be secreting antibodies that are going to be drifting away from it outwards. And we have tons of these pens all next to each other with other B cells that are producing slightly different antibodies. And I'll just draw this in just so it makes some sense here. These antibodies are being secreted by your plasma B cell will also have an affinity perhaps for your tumor cell. So if you could figure out a way to get enough tumor cells, or if you could load the surface receptors onto a bead, you can have your tumor cell present in this. So this is your B cell. And then I'll also have, we'll make it look like a bad guy, this is your tumor cell. And so I will have stuck my B cell into a pen as well as a tumor cell into a pen, and we'll have tumor cells present in all of our pens along with our B cells. And so in some cases these antibodies may stick onto the tumor. So let's make this a really good one in which we've got tons of antibodies from this specific B cell sticking onto a tumor cell. And this is fantastic because if we're able to get antibodies to stick onto a tumor, it's very likely that your immune system can recognize that tumor, and once it's done that, it can effectively kill the cancer that's inside of you before it kills you. So This is a very good pen that we have here. And the unfortunate case is that most of the time the tumor in these other two pens, there's really no affinity for the antibodies that these B cells make for that specific tumor. So these antibodies just kind of drift away outwards, and no one cares about them. So we care specifically about this B cell on the far left here because it has a really high affinity for sticking onto your tumor. And so what's going to happen here is that we're going to make use of a very similar process called sandwich ELISA. And this is somewhat analogous to it, in which we have antibodies from our B cell that have stuck onto some target, and these, the B cell antibodies, I will refer to as primary antibodies, and we will add in an additional antibody into our solution, and we'll let these float around, and these will be secondary antibodies. And I should use a different color, but I'll just describe this process because I didn't think that far ahead. But we have these secondary antibodies that look like this, and these secondary antibodies will have a high affinity for these primary antibodies. And so if your primary antibodies have been localized inside of a pen, because we're able to keep everything inside this pen using the Berkeley Lights technology, we can have these secondary antibodies come in and then stick onto the primary antibodies, and they will contain this little flag thingy I'm trying to draw here. And this little flag thingy is my representation of a fluorophore. And fluorophores are FITC and PE, and there's several other types of fluorophores. The point is behind these fluorophores, and this is a sketch of what the FITC fluorophore looks like. We've just conjugated it onto our secondary antibody. The important thing that we get from these FITC fluorophores, or any fluorophores, is an absorption or an excitation and an emission wavelength. And so as we let time progress in our experiment and we flood our well plates, our quasi well plate, essentially a micro well plate, with tons of our secondary antibodies, what's going to be going on here is that we've got our secondary antibodies that continue to bind into, I'll call this pen 1, this will be pen 2, pen 3. We're going to have tons of our secondary antibodies sticking onto our primary antibodies that are sticking onto our tumor cell, which is very good to us because now we've got a high concentration of fluorophores localized in a region. In this case, it's our pen 1. And because we have such a high concentration of fluorophores present, something really cool will happen when we shine light at a very particular wavelength onto this. And so the FITC fluorophore is sometimes called the 488. You'll see the word number 488 commonly associated with FITC. The cool thing that happens here is that when I shine a 488 nanometer wavelength photon onto FITC, without diving too deep into what is going on with the physical chemistry, you'll have electrons get excited, and then when they relax, they will release a lower energy wavelength photon of 520 nanometers. And what we'll recognize about this is that 488 nanometers corresponds to blue light, what you will perceive as blue light, and 520 nanometers will be perceived by you as green light. And so if we have a high concentration of FITC fluorophores inside one of our pens, they will be absorbing tons of blue light and they will be emitting green light. So if we have a detector for green light, so if I had an eyeball here, and this eyeball could be just a detector that was specific to look at a 520 nanometer wavelength photon, which they do exist and you don't need to worry too much about the technology, but you just look visually at pens 1, 2, and 3, we can see that in pen 1 I have a really high emission of these green photons. These 520 nanometer wavelength photons are coming from pen 1, so that means that the B cell in pen 1 is making some primary antibodies that have a really high affinity for my tumor. And that's really cool because now I have a good idea that, okay, if I take out this B cell here and I sequence its cDNA, the mRNA, its transcriptome, and I look specifically at the genetic material that encodes these primary antibodies, I can start making my own primary antibodies that will actually help me develop a cure for cancer and treat other people's cancers who have the same type of tumor that I do, that's has these kinds of surface receptors on it. And so this is a general introduction to the downstream process that is going on in antibody discovery. Doing a shameless plug for Berkeley Lights, the next cool thing here is that we can take this B cell that we've identified and then export it and sequence it. So this is part of the cool technology that's going on here. And yeah, I hope you guys find this stuff interesting and useful. Let me know if you have any questions, and thanks for watching.